Hello students. So this is our third session where we are going to study about factors affecting enzyme activities. There are many chemical and physiological parameters which can affect enzyme activity, such as concentration of substrate, concentration of enzyme, pH and temperature. Normally what happens, enzymes are adapted to their natural habitats, means whatever the organisms are there, whether it is human, animal or plants or microbes, their body and their structural parameters as well as enzymes produced by them work optimum in that conditions. They are adapted to extreme conditions also where they are naturally living. Okay, but if it is drastically fluctuated, then it happens that activity can be damaged completely. Okay, we will see how pH, temperature, then substrate concentration, enzyme concentration can affect enzyme activity one by one. First of all, we will summarize everything. For example, temperature. Enzyme works at particular temperature range. All enzymes have their own range of minimum requirement of temperature, maximum tolerance of temperature and optimum temperature. What do you mean by that? कि दरेक एंजाइम છે એક મિનિમમ ટેમ્પરેચર જો હોય ત્યારે જ એ એક્ટિવ બનીને કામ શરૂ કરી શકે, મીન્સ કે રિએક્શનમાં પાર્ટિસિપેટ કરી શકે. એક લેવલ આવે ત્યારે એનું જે કેપેસિટી છે, એન્ઝાઇમની કેપેસિટી ઓપ્ટિમમ જોવા મળે. એટલે કે સબસ્ટ્રેટમાંથી પ્રોડક્ટ કન્વર્ઝન મેક્સિમમ એમાઉન્ટમાં થાય. ક્યારે ટેમ્પરેચરે? ઓપ્ટિમમ ટેમ્પરેચરે. ત્યારબાદ એના પછી જેમ જેમ ટેમ્પરેચર વધતું જાય છે એમ એનું રિએક્શન રેટ ઘટે છે. This shows that we are increasing the temperature and production is reduced. Up to what point they can be show their activity like product conversion દેખાડે છે, પણ એક સ્ટેજ આવ્યા પછી જ્યારે ટેમ્પરેચર વધારે હાઈ થશે તો enzyme will denature. This indicates that enzyme activity increases as the temperature increases until it reaches the optimum level. Further increase in temperature decreases enzyme activity. Same way, pH often affects by the enzyme reactions. Means optimum pH, you will find that for particular enzyme activity, 7.2. If for example, 7.2 is an optimum pH. Its activity starts with something like 6 or 4 and till 8 you will find that activity. Okay, same like temperature. You can see in this blue and pink chart that pepsin works optimum around 2, while carboxyl peptidase enzyme works optimum up to 7.8, something nearby that. Okay, means they also have their range. Third is enzyme concentration, effect of enzyme concentration. In order to study the effect of increasing enzyme concentration up to the reaction rate, substrate concentration must be in excess. This means that reaction must be independent of the substrate concentration, so that any variations in amount of product formed in a functional only in the presence of that enzyme. In the same manner, if the amount of enzyme is kept constant and substrate concentration is gradually increased, the velocity or a speed of reaction will increase till it reaches maximum. Any further increase in substrate concentration will not increase the reaction velocity or speed. What does it mean? Means while studying the effect of concentration or substrate or enzyme, one parameter, જ્યારે આપણે enzyme study કરી રહ્યા છીએ તો આપણે substrate નું concentration study કરવાનું છે. જ્યારે એના verses માં if we are trying for a concentration of enzyme, we have to keep the concentration of substrate in a perfect manner, or you can say in a particular amount. Okay. So now, if we go in a little more details, we can see how it happens. Increase temperature. What happens when EV increases temperature? It increases kinetic energy. એટલે કે જે એનર્જી છે જે મોલિક્યુલ્સ ને કન્વર્ટ કરે છે એ પણ ઇન્ક્રીઝ થાય છે. ઓકે, in a fluid this means that there are more random collisions from between the molecule per unit at that time. Okay. The increase in temperature also increase the vibrational energy that molecule have specifically in the case of in this case. Okay. So there will be a strains in inputting, holding the bonds and together. As the temperature increase, more bonds, specifically weak hydrogen and iron bonds, will break and results in this strains. Breakdown within the enzyme will cause the active sites to change their shape. This shapes will less compatibility, and now the denaturation or conversion of substrate into product will decrease. Okay. So this way, in summary, we can say that temperature increases initiation, the rate of reaction with increase because of the increased kinetic energy. However, the effect of bond breaking will become greater and greater, and the rate of reaction will be decreased. Okay, in these two slides, what you have to understand or how you can understand the mechanism. એટલે આપણે જોયું કે જો એક ટેમ્પરેચર પેરામીટર એવું છે કે જે મિનિમમ રિક્વાયરમેન્ટ હોય એની ઓપ્ટિમમ પ્રોડક્ટ કન્વર્ઝન કેપેસિટી હોય એક ટેમ્પરેચરે અને એક સ્ટેન્ડ સુધી એ મેક્સિમમ ટેમ્પરેચરને સહન કરી શકે. હવે આની અંદર મેટાબોલિટિકલી શું ફેરફાર થાય? Metabolically, when the temperature increases, starting માં જે active sites છે એની compatibility substrate સાથે increase થાય અને એ substrate નું product માં conversion easily કરે. એક સ્ટેન્ડ સુધી એ થશે. પછી વધુ ને વધુ તમે જ્યારે ટેમ્પરેચર ઇન્ક્રીઝ કરશો ત્યારે મોલિક્યુલની જે એક્ટિવ સાઇટ છે જે ઇમ્પોર્ટન્ટ છે ફોર બાઇન્ડિંગ ઓફ સબસ્ટ્રેટ એ કેવી થશે? એના એક્ટિવ સાઇટના જે શેપમાં છે એમાં ચેન્જીસ આવશે અને એ કમ્પેટેબિલિટી અથવા કોમ્પ્લિમેન્ટરીટી એની ઘટશે. એટલે જે લોક એન્ડ કી જેવું બનતું હતું એ ઘટશે, એની કોમ્પ્લિમેન્ટરી ઘટતી જોવા મળશે અથવા કમ્પેબિલિટી ઘટતી જોવા મળશે. સો વોટ હેપન્સ કે એક સ્ટેન્ડ પછી જ્યારે તમે ટેમ્પરેચર ઇન્ક્રીઝ કરો છો તો ડ્યુ ટુ ધેટ ઇન્ક્રીઝ ઇન ટેમ્પરેચર કાઇનેટિક એનર્જી પણ ઇન્ક્રીઝ થાય છે જે રિએક્શન રેટને ઘટાડે છે. મીન્સ product conversion ઘટી જાય છે. Okay, this is what we have learned. pH, either acidity and basicity, or you can say acidic nature and basic nature. H plus and OH minus ion are changed, and therefore interference with hydronic or ionic bonds that hold together an enzyme. Since they will be attracted or repelled by the charges created by bonds. This interference cause a change in the shape of enzyme and mainly on an active site. Okay, આમાં બી એવું જ છે, જે bonds છે pH ના changes થવાથી હાઇડ્રોજન અને OH હાઇડ્રોક્સિલ આયન્સ ના bonds નું interference change થાય અને molecule જે છે એ એટલે કે સબસ્ટ્રેટ એન્ઝાઇમ થી એટ્રેક્ટ થાય અથવા રિપલ્ડ થાય છે. Different enzymes have their different optimum pH values, but once it exceed its activity, any activity જે છે એ જેમ તમે optimum pH થી વધારશો અથવા ઘટાડશો તો there will be changes in this H plus and OH minus concentration or its creation of charges, and it will affect active sites, and it decreases its bonding capacity and product conversion. Okay, so this is about pH effect, that it also have a same minimum pH required. At optimum pH, you will find a highest amount of substrate is bounded to enzyme and it converts to product. The same way, when it will be increase or exceed the pH value, it will affect productivity. Okay. Now the concentration of enzyme and substrate also have different effects on enzyme substrate binding reaction, and it is also controlling or regulating whatever the metabolism is occur in a cell. Changing in the concentration of substrate only affect the rate of reaction if it is the limited factor. Okay, this is the one of the thing. In which increase, now we will see that substrate concentration. When substrate concentration increases, it increases the rate of reactions. Because જેમ જેમ substrate વધતો જશે એમ enzyme ને easily available રહેશે અને એ fastly product માં convert કરશે. But once it reaches one level of optimum, then whether you add substrate, it will not affect any productivity. Means the enzyme will become saturated and will be working at their maximum possible rates. Means you can see in this graph, it has gradually increases as substrate concentration has increased. But at this point now, the graph becomes steady. There is no, graph is not going upward direction, means there is no high conversion. Now you can say that now enzyme is saturated by this condition. In the same way, manner, even for an enzyme, when substrate concentration is constant, एक perfect મૂકવામાં આવ્યો છે, તો ત્યારે પણ enzyme એટલો જ substrate utilize કરી શકશે કે જેથી એ enzyme સાથે સારું bonding આપે. એક stage પછી એમાં પણ એનું conversion કેવું થઈ જશે? Steady થઈ જશે. Okay. Now another point before we start this, inhibitors.